In this video, you will only hear my voice and see my fingers address important information as we discuss dihybrid crosses and linked genes. Mendels Law of independent assortment states that when two or more traits are inherited, the genes for those traits are going to assort independently during gamete formation, or meiosis, also called formation of sperm and egg, giving those different traits an equal opportunity of occurring together. So let me demonstrate that for you using fruit flies. In this example, the parental generation, I'm crossing a fruit fly who is wild type for eye shape and wing shape with a fly that does not have eyes, is eyeless, and does not have wings either. So we call that apterous. So our wild fly is homozygous for wild eye shape. So we got E, it's wild superscript for both of those. All right, and this fly is also homozygous wild for wing shape. On the other hand, right, our eyeless apterous fly is homozygous for both of those traits as well. Because they're homozygous for both traits, all of this parental fly's sperm and egg will get one copy of the E gene, of the eye-shaped gene. It's going to get wild from this side, and since the only options here are wild wings, we're going to have that too. Same exact thing happens with our eyeless apterous. They're going to get one copy of each trait in a sperm or egg. Right, so now we have haploid egg and sperm cells from our diploid parents. All right, so if these come together, you're going to get 100% of offspring that are heterozygous for both traits. And since wild eye is dominant over eyeless and wild wing is dominant over apterous, 100% of your offspring will have the wild eye shape and they will have wings. They'll have wild wings, so they'll look just like this parent. However, they're heterozygous for every trait, so their gametes are going to assort independently, and you'll actually come up with 4 different types of gametes for these two traits. A quarter of them will have gotten this eye-shaped gene and this wing-shaped gene, so both wild in this one. Another quarter will have gotten the wild eye and the apterous wing combo, right? The next will have gotten the eyeless and wild wing, right? Whereas the other quarter will have gotten the eyeless and apterous combination. So it's not like the eye gene is always asking, you know, the wing gene, hey, where are you going? Hey, where are you going? Otherwise, right, the F1 gametes would be the same as the parents, and what's the point in that? All right, so what you're going to do then, if you have all these different gametes, you're going to use a 4x4 Punnett square to determine the genotypic and phenotypic ratios of offspring in the F2, because that's a lot of combos. Keeping it classy over here. All right. So, again, here we've got our F1 cross, heterozygotes, 2 heterozygotes for both traits. And if I build a dihybrid Punnett square, putting each of those possible sperm and egg combos that I came up with on the other side of the page along either row and column, and just like any other Punnett square, I drop these traits down, I bring these traits over and see their combination. Then I fill that in. Now if you notice at the top of each square, I've got the genotype, and then below in highlighted, I have the different phenotypes. So anything that's highlighted in blue is the wild eye, wild wing is in pink, eyeless is in orange, and apterous is in yellow. So without having to read all of this, we can break it down using the highlighters to see that 9 out of 16 of our individuals are wild-eyed and wild-winged. 3 of them have the dominant wild-eye feature, yet the recessive apterous trait. Conversely, 3 out of 16 do not have eyes, but do have wings. And only 1 out of 16 they're recessive for both and have both recessive phenotypes. So when you have 2 parents that are heterozygous for 2 traits, the beauty is this 9 to 3 to 3 to 1 ratio you will see all day, every day, no matter how many times you do this square. So if you see 2 parents who are heterozygous for both traits, you don't even really need to do this. You can, but if you memorize this ratio, then you'll be fine. Now, if I make these ratios into decimals, these are our percentages here. So for instance, if you bred parents and had 1,200 fly offspring, the numbers would kind of fall out like this. So double dominant would be about 672. These that have 1 dominant phenotype and 1 recessive one, those would be about 228 each. And again, 1 out of 16 or only 72 flies would show both. However, oh yeah, this is happening. Okay, but that whole 9 to 3 to 3 to 1 ratio only works when the genes for those traits are on separate chromosomes. Because really, if we recall in meiosis, genes don't separate independently, chromosomes do. However, Gregor Mendel, he didn't know that. He didn't know what DNA was. He didn't even know, what a chromosome was. He just figured out genes and looked at the way they were inherited. So we can't blame Mendel. Moving forward. So this only works perfectly if they're on separate chromosomes. So my example is a fly with dumpy wings. Maybe that trait is found on chromosome 2. And ebony body, which is kind of a nice dark brown, okay, that's found on chromosome 3. So again, homozygous wild for both traits. This one is homozygous for the dumpy wings and ebony body. And if this is a parent's diploid genotype, so you can see they've got 2 copies of wild, 2 copies of, 2 copies of chromosome 2, right? So 2 wild alleles, 2 copies of chromosome 3, so 2 wild body alleles, all right? And same over here, you've got 2 copies of chromosome 2, 2 copies of chromosome 3, and the corresponding genes to those. Just so you know, when we're looking at these chromosomes, the little oval in the middle represents the centromere. All right, so individuals from from these parents breed, and their offspring will have 1 chromosome from mom, bam, right? 1 chromosome from dad, right here. So it's heterozygous, it has 1 copy of each gene, right? And then for body, 1 chromosome number 3 from mom, 1 chromosome number 3 from dad. So it's heterozygous for wild and ebony body. Now, see, because these will now assort independently from each other, this heterozygous individual, right, will make 4 different types of eggs or sperm, right? And so they have each of those different combinations that we demonstrated in the previous dihybrid cross. So, hooray! But what if the genes are on the same chromosome? The word for this is called linked genes. Genes are linked, again, if they're found on the same chromosome. So, my example in this case is curved wings and brown eyes. The characteristics, the traits for both of these, the genes can be found on chromosome number 1 in flies. So, in this case, we have a parent who is wild type for both wing shape and eye color. So they're homozygous for both, and both those traits are on chromosome 2. All right. On the other hand, again, we have this brown-eyed curved wing fly right here, and since, oh, I'm sorry, that should be chromosome 2, not 1. Anyway, so yes, these are their chromosomes right there. All right, so their offspring are going to get one copy of the chromosome for each parent. So, right, this parent will contribute one of their chromosome 2s. Bam, with 2 genes on it. And this parent will contribute one of its chromosome 2s. Bam, right here. So, it gets both at the same time. So this is heterozygous for both traits, but its gametes, instead of having 4 different kinds, even though we are looking at 2 alleles for 2 different genes, this organism can only give one set of each chromosome to each sperm or egg. So this one here with both wild traits is going to go into one of the gametes. This one here with both recessive traits is going to go in here to the other half. Right? The chromosomes assort independently, but the genes are linked, so they follow one another. If I mated organisms with these gene combinations, right? So if I mated 2 from this, I would expect my F2 offspring to be 75% wild wing, wild eyed, and 25% to have curved wings and brown eyes. Those would be my expected ratios. Oh, it's happening again. Okay, that works. But alas, reality comes into play. Oh, reality. We must remember that during gamete formation, crossing over can occur, and that kicks this whole thing up a notch. Bam! Okay, so quick review of meiosis, right? So I'm looking here at chromosome number 2 only, right? One chromosome here has a wild for wings and eyes. The other chromosome 2 here has curved wing, ebony body. I'm sorry, brown eyes. And when it's time for meiosis, first thing happens, the DNA replicates. So now I've got 2 sister chromatids for each chromosome hooked together. But during prophase 1, when these homologous chromosomes get together, sometimes the edges of their chromosomes go across each other, right? They cross over and actually switch places from one chromosome to the other. So here I've got the eye genes, the eye color genes on those feet kind of crossing over. Now in this case, instead of just making 2 kinds of gametes, because of crossing over, we have 4. So one gamete carries both wild type genes, just like we would think that it would. And then there's the other one that carries both recessive things, which is normal, and that's exactly what we would expect, right? But then we also get these 2 combinations that we would not expect if these genes were found on different chromosomes. But it's because of crossing over that some little brown eye gene snuck over with the wild wings, right? And then the wild eye then snuck over onto the chromosome with curly wings. So, if we mated these, now the way I've got this going right here, all right, we're assuming that we're going to mate a parent whose gametes, no crossing over occurred, with a parent who did this crossing over situation right here. All right, so that said, we might expect that 5/8 would show both dominant phenotypes. 1/8 would show wild wing brown eye, 1/8 would show curved wing wild eye, and 1/8 would show curved wing brown eye. Now, here's the deal. This is never going to happen in these exact ratios. Now, crossing over, this happens all the time. But these ratios, do not memorize those. I'm just trying to show you that this is very different from that 9:3 to 3:1. And these numbers only exist if only half of these chromosomes cross over in just the right place. But of course, they won't. That's not reality. So let's look at what might happen, though. By looking at the really real reality of linked genes. So we're sticking with our curved wing brown eye example. All right, so with our parent, right, they're both homozygous for the different alleles of that trait. Wilds are dominant over curly and brown, so all their offspring will have wild wings and wild eyes, although they will be carriers for both of the recessive traits. If my flies mate and have 1,176 baby flies in the F2, if these are unlinked genes, we would expect typical independent assortment to come up with that 9:3 to 3:1 ratio. That's what we would expect. And so if we had 1,176 flies, those are the expected values that I put in there. However, perhaps in fact, we would observe these very different and strange numbers. So instead of 75% wild wing wild eye, 25% curved wing brown eye, and 0 curved wing wild eye and wild wing brown eye, like if we looked back at that 75% thing, this is if we had 9:3 to 3:1, if it were assorting independently. Anyway, basically we see these guys who should not be there if these genes are, oh man, I'm gonna have to definitely stop and revise this. Okay, timeout. Okay, so if these were unlinked, we would expect this nice 9:3 to 3:1 ratio. But what we observed is the vast majority being wild wing wild eye, and another good bit being curved wing brown eye. All right, and then you have this little smattering of other phenotypes, right, are combinations of one recessive and one dominant. So what these numbers show us is that A, they're probably linked because this number and this number look a whole lot like 75%, 25%, but not only are these linked, but crossing over has occurred, otherwise we wouldn't have been able to get these small groups of flies. So since these expected values and our observed values are not even close to the same, we can conclude that the allele for curved wing and brown eyes are linked together and that crossing over has occurred.